Now let's start thinking about how epigenetics is linked to cancer. Now let's remember that traditionally, cancer was thought to be caused by gene mutations, often environmentally induced gene mutations, but still gene mutations. And this was pretty much the dogma of cancer. Genes get mutated, enough of them get mutated, and cancer forms. But as we bring in this idea of epigenetics with cancer, what we're saying is that cancer can be caused by, or at least influenced by, a non-mutation pathway. And one of the early studies looked at colorectal cancer. And in these colorectal cancer cells, many of the CPG islands were hypermethylated. Hypermethylated, of course, means that there are more methyl groups on here. And if you remember, hypermethylation of the CPG islands means a decreased expression. Hypermethylation reduces the expression of some genes, so this was quickly linked to tumor suppressor genes, that when they have an effect that leads to cancer, it's because they are no longer expressed. And sure enough, these colorectal cancer cells, as well as many other cancers have now confirmed it, cell cycle genes like retinoblastoma, DNA repair genes like BRCA1, that's the one that's most often linked to breast cancer, genes linked to apoptosis. One of these genes is called TMS1. Genes linked to hormonal regulation, like the gene called ER for estrogen receptor. Genes linked to metastasis, like CDH1, which is a breast cancer linked gene. And then angiogenesis, and this gene is TSP-1. All of these genes in various cancers, including colorectal, but also breast cancer, prostate cancer, and several other cancers, all have these genes hypermethylated. The genes themselves aren't mutated, the promoters aren't mutated, but rather the promoter regions are hypermethylated, thereby decreasing their ability to be transcribed, and therefore decreasing the likelihood that they will be able to stop the cell cycle when they need to. Now we'll talk about hypomethylation on the next podcast. Let me squeeze it in here real quick just so we have some information on it. So hypomethylation is also seen in some cancers. It's not quite as predominant, at least not that we know of, but it still is seen. And this is linked to converting proto-oncogenes to oncogenes. A classic example is c-Myc, and c-Myc is the same gene that we talked about when we talked about the iPS cells. But this is an oncogene that helps drive the cell cycle. And the promoter regions of these c-Myc genes in some cancers are hypomethylated. Being hypomethylated, these genes would be transcribed, and in this case, in an unregulated fashion. So let's spend a little bit of time talking about hypermethylation. It is thought that this is caused by a overexpression of DNMT, the DNA methyltransferases. Or it could be caused by aberrant recruitment. So it might be recruited to genes it's not supposed to be recruited to. Another thing that could happen is that these DNMTs, instead of being overexpressed, have a point mutation. And this point mutation is just enough to make it fold a little bit differently, so it has a new specificity to DNA. It would bind to DNA more efficiently and thereby methylating it more efficiently. What they've discovered is that in many cancers, we see this hypermethylation, but we don't just see this hypermethylation on just one gene. Often it occurs on multiple genes. And we call this the CPG island methylation phenotype. And for short, we call it CIMP. So again, this is when multiple genes in cancer cells are hypermethylated. CIMP is characterized by excessive DNMT1 expression. And not just expression, but also activity. Wouldn't matter a whole lot if you're expressing a bunch of it, but it's not active. So it's excessive DNMT1 expression and activity. So we're making a lot more DNMT1, and it's more active. Why doesn't the cell regulate this anymore? All the cells are doing is making more of this enzyme. Why can't it regulate it the same way it would regulate if it was just one copy being expressed? Well, we don't really know, but some ideas are that its expression is so high that it can methylate non-CpG islands. It's also possible that the transcription factors that are involved in recruiting or not recruiting DNMT1 no longer can regulate DNMT1 because there's too much of it there. It has also lost the preference for hemimethylated DNA. DNMT1 usually prefers hemimethylated DNA, but it's not exclusive. It will methylate other regions as well. And when you have so much of it present, it's more likely to methylate naked DNA. That is DNA that does not have methyl groups already on it. So now I want to list a few experiments that were done with DNMT1. The first is when we mutate DNMT1 in cancer cells. The cancer cells go through apoptosis. So DNMT1 would be predicted to be important in maintaining these cancer cells, because when we get rid of it, the mechanism to get rid of that cancer cell is reestablished. Now, opposite this, when we overexpress DNMT1 in healthy cells, they are transformed into cancer cells. It can then be reversed by adding in small interfering RNAs. Those RNAs that bind to the messenger RNA and direct it for destruction. So you overexpress DNMT1 and you get cancer. You remove that expression by getting rid of the messenger RNAs of DNMT1 and you revert back to healthy cells. Now, it's unlikely that DNMT1 acts alone because we know there were other proteins that had to be present on the chromatin to recruit DNMT1 or to prevent DNMT1 from being there. So it's unlikely that DNMT1 is acting as a lone protein in transforming normal cells, healthy cells, to cancer cells. Now, if you remember when we talked about learning and memory, we said that a general phenomenon is when people age, that results in less methylation. However, in cancer cells, methylation goes up. It's hypermethylated. So how does this happen in cancer cells? Again, we don't know for sure, but some ideas are that there are other proteins involved. This seems almost certainly, as I just mentioned, in the cancer cells, which make them different than a healthy cell that's just aging. This overexpressed DNMT1 or mutations in it that change its affinity will bind that naked DNA more efficiently. DNMT1, interestingly, will help drive the cell cycle into S phase. It does so by activating certain genes, but in this particular case, as it's linked to S phase, it's not because it methylates the promoter or removes methyl groups. It's an independent process of DNA methylation. And another way that overexpressing DNMT1 can lead to cancer is that DNMT1 can bind other tumor suppressor genes or proto-oncogenes. In this case, I'm going to talk about how DNMT1 can bind to p53. p53 is one of the known master regulators of cell division and ultimately to cancer. And when DNMT1 binds to p53, together they regulate genes linked to p53 expression. We know that when p53 is expressed, it can fix the genome, it can signal the cell for apoptosis, it can do lots of things. But in many of these instances, p53 will bind to DNMT1 to help regulate this expression. And when DNMT1 is expressed at a high level, this can lead to a misregulation of p53. Okay, that's all I have for this podcast, linking cancer with epigenetics. In the next podcasts, I'll talk about hypomethylation and some other aspects of epigenetics and cancer. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.
You're still here? It's over. Go home. Go.